Hey guys, I'm actually going to go through today's notes with you. I really think it's important that I do that for you to understand how to do the worksheet that you're going to be doing in a couple days or tomorrow. These notes that I have on my screen right now, these are actually notes that you should have taken last week. I believe it was last Friday. And there's actually just a couple things I really feel that I need to point out on this before we do today's notes, that will ultimately help you with the activity you're doing tomorrow and eventually next week too. So I'm not going to go through this whole presentation, just a couple things that I want to point out on it. So this presentation was really about sort of the background of DNA, how it's put together, just some really important basic information that will help you with the future things that you're going to be doing with DNA, and again, the future labs that you're going to be doing there. So, again, I'm not going through this whole presentation because you should have already taken these notes, but I do want to point out this background picture here. You have a piece of DNA. This shape of DNA is called a double helix. It's kind of like a twisted ladder where you have this pink ribbon on this side, and then you have another pink ribbon on the other side. So it's two-stranded. That pink ribbon is made up of sugars and phosphates, and then down the middle, kind of the rungs of the ladder, you have these bases which they've color coded, purple and yellow, and this salmon and this green color there. And you'll notice that it looks like the salmon and the green always go together, and the purple and the yellow always go together. And that's an important thing that you're going to see in a couple slides. So I'm just going to kind of zoom through this part. DNA stands for deoxyribonucleic acid. This is just a little bit of the brief history of discovering it. Important jobs that it needs to do. It carries your genetic information. It has to be easily copied. This is what I want to focus on a little bit here. So it's really long. The human DNA, a strain of human DNA, is about a meter long. And for you non-metric individuals, that's roughly 3 feet. And again, it's made of two sides. Each one of these sides, it's built with monomers called nucleotides. So a nucleotide has three basic parts to it. It's got a sugar. In the case of DNA, that sugar is called deoxyribose. It's got a phosphate. And the sugar and the phosphate is what makes up these, the sides, these twisted laddered sides that you have there. And then down the middle, you have these bases, and there are 4 bases found in DNA. You have adenine and guanine. We usually just use A and G for short. And you have cytosine and thymine. We use C and T for short because it's just a lot easier to write A's and T's and C's and G's. And again, this backbone, this pink strand that they have in this picture, is sugars and phosphates repeated. And you can string together these A's, T's, C's, and G's in an infinite number of ways. The human genome is about 3 billion letters long, so you can have an infinite number of ways that you would want to, or that you could put those letters together. Here again, it's just showing you one side of the DNA. It's kind of on its side, and there's your adenine and guanine. Notice they're bigger, and then you have cytosine and thymine. Notice they're smaller. This is your sugar, this little pentagon, and there's your phosphate group there. So this is one side of the DNA strand. I really like this diagram. You may very well see it come up again later. Hint, hint, wink, wink. They've outlined what a nucleotide is. So there is your base. There is your sugar. There is your phosphate. These blue strands are your sugar phosphate backbone. And then they've got your bases down the middle here. And notice they've got them drawn kind of sciencey chemically, and then they've just got them drawn by shapes. They have a color code over here. Notice that the C's and G's are going together always. They could be on opposite sides. Notice the C is over here on the left and this part of the picture, and then over here it's on the right, and then notice it's flipped. That's okay. But notice that the C's and G's always go together and the A's and T's always go together. There's 2 reasons for that, 2 big reasons for that. One is the shape has to fit, and they're showing you that here. See how the C and the G, they plug together, and the A's and the T's, they plug together? You couldn't put an A with a C, it wouldn't fit. Now, granted, they don't have these exact shapes, in real world, but they do have a 3-dimensional chemical shape. So they have to fit together. And they're showing you that here with these shapes. The A's and T's are curved, and the C's and G's have these points. Up here, they're showing you hydrogen bonds. See how the C's and the G's, they require 3 hydrogen bonds? Well, the A's and the T's only require 2. So a C requires 3 bonds. It couldn't go with a T because T's need 2 bonds. So the number of bonds has to match and the shapes have to match. So that's 2 ways that DNA prevents the wrong bases from hooking up the wrong bases. And that's in this diagram here. So that's one reason why I really like that diagram. So there's a base pairing rule where A's can only go with T's and G's can only go with C's. And instead of remembering these really long sentences, there's a tool to help you remember that. And this is the tool. And it reads A2T G3C. That's not an equal sign. It's A2T G3C. And what's really nice with this is it rhymes. A2T G3C. It's short. A2T G3C. But it also has one of the reasons why you can't mix and match the letters. And that's right here. This double hash and this triple hash, whatever you want to call it, this represents the number of bonds that have to happen between the A's and the T's and the G's and the C's. So A's and T's require 2 bonds, G's and C's require 3 bonds. It's not showing you the shape. That's the other reason why you can't mix and match, but it does give you one of the 2 reasons why you can't mix and match the letters with that. So now that you have this base pairing rule, if I give you one side of the DNA, so here's one side, remember DNA is 2-sided, and I have this kind of flipped on its side. If I give you one side of the DNA, so here's just one small sample of DNA, with that base pairing rule of A2T G3C, you can do the other side. So you only need one side of the DNA, and using that tool, you can get the other side. So if you think about it, G's go with C's, right? T's are going to go with A's, C's go with G's, A's are going to go with T's. So if you look at it, right, so what's going to go on the other side of the G? A C. What's going to go on the other side of this G? A C. What's going to go on the other side of this G? Another C. Another C. What's going to go on the other side of the T? An A. An A. Here we have a C. What goes with C's? G's go with C's. So you can just keep doing that, and here's your answer here so that you can see. I only need to give you one side. You can figure out the other side as long as you remember the base pairing rule of A2T. So A's and T's go together, and G's and C's go together, G3C. So you could put the bonds in here if you wanted to. 3 dashes would go between the G's, and 2 dashes would go between anywhere you see an A and a T, right? So these were the notes that you took last week. I just wanted to go through especially this A2T G3C thing and being able to do this. If you get one side, you can do the other side. That was really important. So that was last week's notes. There. So I want to talk about today's notes. So today's notes are all about something called RNA. Okay, so RNA, let me get this to slideshow so that we can see it. So RNA is a little different from DNA, but it also has some similarities. So RNA, as you'll notice here, this is RNA. How do I know this is RNA? Well, there's a couple big things. One, it's single-stranded. DNA is double-stranded. But there's also U's here. Notice there's no T's in this. There's U's instead. That U stands for uracil. So U's are uracil. Notice there's no T's. Notice, though, there's still C's and G's and A's, but there's no T's. So this is a molecule called RNA, or ribonucleic acid. So this is today's notes, and I felt that these notes are really important to go through with you because, again, they're going to help you with the upcoming things you're going to be doing. So, again, genes code for your proteins. Those proteins determine your traits. So, genes indirectly code for proteins. Directly code for proteins and indirectly control your traits. Proteins are what control your traits there. So, DNA codes for proteins. Those proteins determine your traits. So, all along you've been told, DNA determines your traits. And yes, that's true, but it's kind of indirectly. DNA codes for proteins first, and those proteins determine things like your eye color and your hair color, how tall you're going to be, and how fast your metabolism is going to be, and that kind of thing. So DNA, if you remember, DNA is found in the nucleus of cells. And DNA is obviously incredibly important, coding for these proteins which determine your traits. And if something happens to this DNA, you can have things like cancer result, and cells controlling, cells dividing uncontrollably, and all that stuff. So DNA is incredibly important that it's kept safe. So DNA is locked, like in a filing cabinet, locked in the nucleus. So the problem, though, is proteins are made where? On ribosomes. So how do we get the instructions for making a protein from the nucleus, that's where the DNA is, to the ribosomes, which are out in the cytoplasm, without letting the DNA out? It's kind of a conundrum there. So what we have instead going on in our cells is we have something called RNA, or ribonucleic acid. RNA is going to be kind of like the middleman. He's going to be the messenger. RNA is going to be the messenger that brings the instructions from the DNA in the nucleus to the ribosomes in the cytoplasm. The RNA, if it gets damaged, it's not a huge deal because we can always go back and make another copy of it from the DNA in the nucleus. But if the DNA gets damaged, then every copy we make after that, every piece of RNA we make after that is also going to be damaged. And now you can have situations where proteins aren't made, and then those parts of the cells may not get the instructions that they need, and then you can have cell death or cancer or all sorts of things. So what we have instead is we have RNA that's going to bring the instructions from the DNA, because again, the DNA can't leave the nucleus, to the ribosomes in the cytoplasm where those proteins are going to be made. So RNA, it's got some similarities to DNA. It's got a 5-carbon sugar, different sugar. It's ribose instead of deoxyribose. It's got phosphates, and it's got bases, 3 of which are the same as DNA. But there are some differences. Again, the sugar is different. It's ribose instead of deoxyribose. It's single-stranded. DNA is double-stranded. And RNA, again, has uracil instead of thymine. So in RNA, you can still use the tool, the A2T G3C thing. However, with RNA, it's A to U G3C. So the U thing is a huge way to know that you're working with RNA, that messenger, that disposable working copy of DNA, right? So the base pairing rule, it's the same. It's just with RNA, you've got to put U's instead of T's. There are no T's in RNA. So that's a huge clue to figure out if you're working with DNA or if you're working with RNA. Look for the U's. If you're working with U's, you're working with RNA. If you're working with T's, you're working with DNA. Types of RNA, there are actually 3 different types of RNA. Again, remember ribosomes, that's where proteins are built. So there's messenger RNA. That's the RNA type that I'm talking about right now. Shorthand, we write mRNA. This is what's going to bring the message from the nucleus to the ribosomes. And there's another little picture of it. Notice uracil. They have it in brown, and they got a little notch on it on this one. There's also ribosomal RNA, or rRNA. This is a type of RNA that helps make ribosomes themselves and also helps with the process of making proteins. And then we have transfer RNA, or tRNA. tRNA looks like a T. It's got this little section on the bottom of 3 bases that we're concerned about, and on the top is where what an amino acid would be, and there's 20 different amino acids in humans. tRNA is going to come up in another presentation later after break, I believe, to help you with learning a little bit more about tRNA. So a little flow chart here that you could feel free to go through, at your pace. So how do we make RNA? Well, earlier this week you went through a presentation about making copies of DNA. Making copies of DNA is really easy. As long as you have one side, you can make the other side. But this presentation is talking about, well, how do we make the mRNA? How do we go from DNA? How do we get the instructions from DNA into that messenger RNA that we can then send to the ribosomes in the cytoplasm? So that's a process called transcription. The prefix trans means change, and scription refers to write. So we're rewriting. We're going to rewrite the letters of DNA, A's, T's, C's, and G's, into the letters of RNA. We've got to make a copy of DNA into RNA so that we can send that copy to the ribosomes, right? So this is happening in the nucleus because that's where the DNA is. And what we're going to do is we're going to take a copy. We're essentially going to take 1/2 of the DNA strand. Remember, it's 2-stranded. We're going to take 1/2 of the DNA and we're going to copy that into what's called a complementary sequence of mRNA. So we're going to take one side of the DNA and we're going to rewrite it into, like, essentially making the other side of DNA, like I just did a little earlier in this video, but we're going to do it with RNA letters. So it's the same exact thing I did at the beginning of this video, except wherever your brain wants to put a T, if you're making RNA, you've got to put a U because there are no T's in RNA, right? So this is transcription, okay? And we're going to use an enzyme called RNA polymerase, very similar to DNA polymerase in the process of DNA replication. It's just a slightly different enzyme that helps make RNA. So during transcription, you're going to take this RNA polymerase. Again, it's similar to DNA polymerase. It's essentially going to help unzip. There's other enzymes too, but just to break it down, you're going to unzip the DNA, and you're going to have this RNA polymerase that's going to come in and bring in the correct bases depending on the side of the DNA that you're transcribing from. So that's what they're showing you here. This purple wiffle ball thing, that's the nucleus. There's your RNA polymerase unzipping the DNA. It's using one side of the DNA, this blue strand, to help make a copy, a working copy of that set of instructions. In this case, it's making it into RNA. A little hard to see in the picture, but they do have uracil as the darker orange and thymine as a lighter orange. So RNA, again, you're going to have U's instead of T's. So once we have that piece of RNA made, it's going to leave the nucleus. So it's going to leave, this RNA strand is going to leave through one of these pores, these nuclear pores in the nucleus, and it's going to go hook up with a ribosome. The process of taking the mRNA and turning it into proteins is called translation. So transcription is rewriting letters. Transcription is rewriting the letters of DNA into the letters of RNA. Translation, remember, trans means change. Lation refers to like language. So we're translating. We're turning it from one language into another. We're going to turn the language, the instructions of mRNA. We're going to turn those instructions from mRNA, the language of mRNA, into the language of proteins. And the language of proteins is amino acids, right? So we're translating here. Remember, proteins, which are made of amino acids, proteins code for your traits. And we're going to string together amino acids in an infinite number of ways to make these polypeptides called proteins. Okay. As I mentioned a little earlier, there's 20 different amino acids in humans. Our bodies make about 10 of them. The other 10 or so we have to eat. We have to get in our diet. So that's why a proper balanced diet is so important. The order of those amino acids, the way they're put together, determines what proteins will be made. And the language of mRNA, those sequence of letters, the A, the U, the G's, and the C's, which we get from copying from a strand of DNA, that's what's called the genetic code. And again, we're going to be working with A's, U's, C's, and G's here because this is RNA. Okay? This part here is really important because it tells you how to read the genetic code, how to read the RNA. So we read RNA 3 letters at a time. So like AUG, ACC, GGA, GGU. We read them 3 letters at a time, and that's called a codon. One codon codes for one amino acid in a protein. So again, you have 3 letters, AAG. That's a codon. That codes for one amino acid. Which amino acid? There's a way to figure that out. You don't have to memorize it. So here's an example. Here's your mRNA sequence, right? We read it 3 bases at a time. So I broke it out with these dash, but you don't have to. When I'm doing this on paper, I tend to put a nice big giant slash here, a nice big vertical line. But I broke it out. There's 3 codons here. UCG is a codon. CAC is a codon. GGU is a codon. Each one of these codons, which is 3 letters long, codes for one amino acid. In this example, UCG codes for serine, CAC codes for histidine, and GGU codes for glycine. Now, you don't have to memorize this. There's a tool to help you with that. But let's just double check something. So if this is the mRNA sequence that we transcribed, right, what's the DNA sequence we got that from? Well, it's essentially just doing the other side. But you got to remember, this is asking about DNA. DNA has T's in it. Wherever you'd want to put, a U, you'd have to put a T if you're working backwards. This is essentially working backwards. I'm asking you to go backwards. So, U's go with A's. So this would be an A. C's and G's never change. So you'd still have a G here, a C here, a G here. Now this is tricky. What would go on the other side of the A? Well, for DNA, it would be a T. Then you would have G, C, C, and you'd have A there, just to show you I'm not lying. There it is. So if I give you an mRNA sequence, you can go backwards. You can go backwards to figure out what the DNA sequence is. You just got to remember with DNA, you've got to put T's. So practice this. Even if you just practice the one on the screen, practice that. So because there's only 20 amino acids, you're going to have some overlap with these codons. There's actually 64 codons. There's 64 different ways you can put together those 4 letters into codons. So you're going to have some overlap. So some amino acids will actually have 6 codons that code for them. And some will only have 1. There are 3 what's called stop codons. Those tell the ribosome they're at the end of a protein. There's no amino acid that codes for those codons. There is one codon you should memorize out of the 64, and that's AUG. AUG is a very special codon. It codes for an amino acid called methionine. AUG codes for the start of a protein. So that's how the ribosome knows where to begin. It's going to scan until it reaches AUG. Once it reaches AUG, it knows now it can start putting together those amino acids to make the protein until it reaches one of those 3 stop codons and there's no amino acid coming. It will then release the protein and it will go do what it needs to do. So how do you figure out which amino acid you're working with? Well, there's 2 different charts you can use. I can almost guarantee you're going to like this first one better than the second one. However, this first one with this circle thing, I've never seen except in one textbook. But I can almost guarantee you're going to like this one better. So notice there's sort of, there's 4 rings, I guess. But you have this center ring here, the center circle. Then you have this sort of middle circle, which is a slightly darker yellow. And then you have this third circle, which is even darker yellow. And then you have your amino acids kind of on the outside here. So how do you do this? Let's say that your mRNA sequence, your codon is UCG. How do you do this? You start in the center. Find your first letter of your codon, U. So now I know my answer for what amino acid this codon UCG codes for is in this quadrant, this part of the circle. Then I go to the second ring and I find my next letter, C. Okay, so I've narrowed it down, right? I know I'm in this sort of chunk here. Well, any other letter is going to code for serine, but it happens to be G. We can double check it's serine. So UCG codes for serine. And just to show you, UCG codes for serine. Now, if I back this up, so does UCA codes for serine. UCC codes for serine. UCU codes for serine. So that's what I mean by you can have multiple codons code for the same amino acid. So our first amino acid is serine. And that, okay? How about CAC? Again, C, right? C, A, C codes for histidine. If you had C, A, U, that would also code for histidine, but this is CAC, it codes for histidine. So our next one is histidine. Okay. Try the next one. G, U. What's that code for? Glycine, right? So that's how you use this. Now, I'll do another one with you. Try CCC. So C, codes for proline, right? Try another one. UGA. What's that code for? So UGA. That's one of the 3 stop codons. If you just scan up a little bit more, you'll see the other 2. UAG is another one, and UAA is another one. There's that AUG that I told you about, methionine. That's your start codon. That's the only one I would bother memorizing. It'll just speed things along for you. You also have this chart. Now, this chart is the one that you're going to find pretty much in any textbook or online. Like I said, that circle one, which I actually like better too. The circle one I think you're going to like a lot better, but I want to show you how to use this one as well. So let's use the same codons as before, UCG. Notice on this chart we've got first letter on the side, second letter across the top, and third letter across this other side on the right side here. So how you use this is you find your first letter, U. Okay, so there's U. I know my answer is somewhere in that red rectangle, somewhere in this top row, my answer for what amino acid UCG codes for. My second letter is C. So I look across the top, there's C. So now my answer is somewhere in this blue box, somewhere in this section. And then I find my third letter over here, which is G, right? UCG is right there. Now again, anything in this box is going to code for serine, but you could double check it. There's UCG codes for serine. Okay? So serine is our answer for that one. I'm using the same codons as before just so that you can see you can use them either way. C, again, your answer is somewhere in that red rectangle, somewhere in this row. Second letter is A, right? So you follow it down to somewhere in this blue box here. And then C, third letter, follow it over. CAC is histidine with that one. So that's how you use this one. Again, I'm going to guess and say you're going to like the round one better on the previous slide. GGU, G, Answer is somewhere in this red rectangle in the bottom row. Second letter G, right, up top. Follow it down. It's somewhere in this blue box. And then you have U over here. That codes for glycine. Anything in this box will code for glycine, but you can double check it. GGU right there. So that's how you use these charts. And you're going to need these charts for upcoming things that you're going to be doing, like tomorrow or the next day, whatever the next day of school is. I have a worksheet for you to work on this. And then there's also a lab that you're going to be doing with this too. So don't forget, these two things are here in this presentation. You might even want to print one out if you don't get a copy, whichever one you prefer to work with, whichever one's easier for you. So that's this presentation. If you ever have any questions, please feel free to shoot me an email. I am checking my email, and I will get back to you as soon as I can. Okay? Hopefully this was helpful. Bye, guys.